Thank you very much, Rachel. So hello everybody, and thank you for coming along today on this lovely wet windy autumn morning. So today I'd like to tell you a little bit about my PhD research, where I looked at approaches to managing the tree disease ash dieback. And more specifically, I looked at whether soil-related factors influence the tree disease and whether the use of sustainable soil amendments could have a positive effect for the ash trees. So the common or European ash, Fraxinus excelsior, it's a very common tree. You're likely to see them in hedgerows, parks, gardens. In fact, it's estimated that before ash dieback, there were around 90 million ash trees in the UK. And the tree is easily recognisable by its pinnate form. It is a compound leaf made up of leaflets on a central vein, which is known as the rachis. Now, the tree is native to temperate northern Europe and it was recognised as a tree that was actually going to do quite well out of climate change. And it suffered from very few pests and diseases. That was until ash dieback came along. Now, the ash dieback disease is caused by an ascomycete fungus. An ascomycete is a tiny little cup-shaped fungus. And you can see in this picture here, this is the midrib of the ash leaf, magnified many times, and it shows how tiny the fungus actually is. So Hynocyphus fraxineus, the ascomycete fungus, is an introduced species. It's not native to Europe. It's believed it originated in northeastern Asia, so the Manchurian area of China, and it was accidentally introduced into Europe. It's thought on ornamental trees. Initially, it was thought it was in the 1990s, but we're now starting to think that the fungus has been here a lot longer. And since then, it has started spreading across Europe, and it reached the UK around the 1990s, although again, it's likely it came here earlier. It was imported on nursery stock, and this was after we knew about the problem on continental Europe. So it highlights the importance of phytosanitation, that we don't accidentally bring in more invasive species. The tree disease did actually also come in on spores that blow in across from the continent, where it's a wind-blown fungus. So the fungal pathogen, all going too well, isn't it? Okay, how do we put that in prevent mode? Hopefully people online can still see that. Okay, so the fungus, its life cycle is such that it produces spores that are borne on the wind. So in the summer, the spores land on the ash leaf where they infect the tree. They cause little brown necrotic spots, and the fungal hyphae then start to spread through the leaf, down the veins, and into the midrib, the rachis. The leaves then start to fall off the tree, and the fungus lives in that central midrib where it creates a protective layer around itself, lands on the woodland floor, and it lives in there quite happily over winter, protected from desiccation and other microbes. Then in the spring, it starts to produce those little cups, the fruiting bodies that we saw in the previous picture. It then produces its spores in the summer, which are borne into the air and then land on the leaves of the tree, reinfecting the ash tree. So here we can see some images of ash trees with dieback, and we think sadly we're probably all quite familiar with what dying ash trees look like. If we go past the hedgerows round here, you'll have seen twiggy looking trees, and they're most likely ash trees. But the fungus gets into the leaf and it stops moisture being able to get in. And that causes the leaf to turn brown and start to wilt and droop. So this is a young sapling. Then some of the fungi start to spread into the woody tissue of the tree, and that's when the problems start to happen. The fungus blocks the conductive tissue within the stems, and moisture can't get beyond that point. We start to see these diamond-shaped lesions forming on the tree, and the tree becomes quite dry above that point, and we get this characteristic cinnamon brown colour. And eventually, as the disease takes hold in the woody tissue, the tree starts to die back, and we get these trees that have very, very few leaves on them. Sometimes the ash tree can stage a bit of recovery, and it puts out what's called epicormic growth from the trunk and the branches. It puts out loads of leafy growth. The tree starts to look a bit sort of bothery, and you might think that the tree is starting to recover, but unfortunately, that is just a big symptom of the disease. Now, it was estimated that of the ash that were in Europe prior to the disease, only around 1 to 8% have any tolerance of the disease at all. So we're likely to lose a very large number of trees. However, more recently, researchers have found that naturally regenerating ash are starting to show more resilience, and it's likely that they will be better able to survive. So there is some hope if we can protect that young natural ash regeneration. Lovely, thank you. So thinking about soil-related factors and ash trees, so soil, of course, affects plants, and the ash tree is no different. But the soil conditions, the conditions on the ground, because the fungus is in that central leaf midrib that lands on the floor, the fungus is also affected by ground and soil conditions. And studies have reported that ash growing in soils with higher levels of organic matter have more severe ash dieback. And ash trees that grow on neutral and alkaline soils also have worse ash dieback symptoms. But overall, there's been very little research into the effects of soil on this tree disease. And most of them have looked at proxies for soil factors. They've used indicators and indicator species rather than actually testing the soil. No, yes, no. Yeah. Looks for a phrase.
Maybe it's the bottom left that was presented on the upper. It says 06 to 27. There's a
Just used it.
yes.
Yes, okay. So the other factor that has a big influence on ash dieback is soil moisture. So symptoms are more pronounced in very waterlogged soils, and there have been positive correlations observed between levels of soil moisture and severity of the disease. It's likely that the soil moisture conditions create more humid above ground conditions, and that the fungus, when it's there on the ground, can sporulate more, produces more spores, and that then creates more disease pressure on the tree. So damp conditions are also beneficial for other fungal pathogens. You might have heard of the honey fungus, armillaria, and that attacks trees that are already quite stressed. So soil moisture is indeed playing an effect. So
You might just be able to use the forward arrow.
yeah. So the other thing that came to mind, I wanted to look at what
Extension
music day. Okay, so gosh, okay, so we wanted to see whether taking an agroecological approach might help the trees, because soil amendments, things that we add into the soil, have been found to have a beneficial effect, but most studies have been on annual crops. But do these soil amendments work for trees? And I think because trees are generally less focused on by researchers, they're much bigger, they grow much more slowly, and they're probably less commercially viable. There's been a lot less research. But the soil amendments do improve outcomes for plants. So I wanted to test whether this was the case for trees and ash dieback. So the soil amendments we looked at were compost, a peat-free compost, wood chip mulch, so willow in this case, biochar, and biochar combined with comfrey tea. Okay. So what actually is biochar? For anyone who hasn't come across it before, biochar is quite simply charcoal, the stuff that's cycled on a barbecue. But when it's used in an agricultural or environmental context, we refer to it as biochar. And it's made when any organic matter is brought to very high temperatures, usually above 250 degrees, in the absence of oxygen. So it's not combustion like you get with fire because there's no oxygen. Instead, it's pyrolysis. So biochar can be made from a huge range of different feedstocks, from woody material, crop residues, through to things like sewage and manure wastes. So the feedstock, the material the biochar is made from, influences the properties of the biochar. So the feedstock and pyrolysis temperature cause a big change in the material. And biochar usually has quite a high pH compared to the underlying feedstock. And it also has a structure made up of millions of nanoscopic pores. And those features are really important for how it interacts in soils. So it's quite challenging because, as I said, if you've got lots of different feedstocks, the properties of the biochar that emerges can vary considerably. And there is a school of thought, as with compost, that we don't refer to a single biochar, but we consider it as biochars in the plural. So depending on the type of biochar and how it was made, it will interact differently when we add it into the soil. But soil isn't a homogeneous thing. Soil varies considerably, its texture, its bulk density, its pH, and so the biochar will interact very differently depending on the soil to which it's added. Those combined then have to interact with the plant, and studies have shown that biochar interacts very differently with different plant species and even different genotypes. So we're likely to see a big difference looking at angiosperms, so broadleaf deciduous trees, and conifers. The different mycorrhizal interactions in the soil are likely to have an effect. Then we also have to consider the type of pathogen when we're looking at a pathosystem. So is it a fungi? Is it a bacteria? What are the trophic habits of that pathogen? What will they eat? Do they eat living tissue or dead tissue? So all of these factors influence what's going to happen. And we've even found that the results from pot trials using exactly the same materials can differ considerably to biochars being added to soil, probably because there's movement of microbes and a lot more going on in the bulk soil. So we need to take all of these factors into consideration. But perhaps the most important is the role of soil microbes, and it's likely the effects of biochar are mediated by soil microorganisms, so it's not necessarily a direct effect on the plant. And this comes about because biochar is incredibly rich in carbon, and many soil microorganisms consume carbon, they're copiotrophs. So adding carbon in this form into the soil might be providing food for them. It also creates a safe habitat. So when we think about all those little microscopic pores, the fungi and the bacteria can get into those pores and keep away from predators that would otherwise eat them. The pores also mean biochar can take up and absorb compounds within the soil. So it might be taking up microbial signalling molecules, and this is likely to alter the balance between different fungi and bacteria in the soil. And biochar's pH, as I said, it usually has quite a high pH. That is likely to favour bacteria over fungi that prefer more acidic soils. And some biochars can add toxins into the soil, so things like volatile organic compounds, PAHs, and things like that that might harm some microbes. And all of this is likely to change the diversity and abundance of microbes, and in particular, the plant growth promoting rhizobacteria and fungi, Pg, Pr, and F. And these are microbes within the soil that colonize the plant roots and interact with the plant. And in doing this, they trigger the plant's hormone signaling systems, and that can improve plant growth, but it also primes the plant's defense responses. And that can help the plant be ready to fight against any infections across the whole plant. So it's not just happening at the roots, it's also happening systemically in the leaves and all parts of the plant. So research into biochar is not a new thing. People have been looking at the potential for benefits in plant growth and in plant disease suppression since back in the 1800s, in fact, possibly even earlier than that. And studies looking at trees were going on in the 1940s. But the real interest in biochar and its potential for soil fertility arose in the 1960s with a guy called Wim Sombroek, who went into the Amazon and found these patches of very dark soil that was very, very fertile and retained its fertility for thousands of years. And it's believed now that these soils built up over time with inputs of kiln fires, human waste products, food, plant material, and that they evolved over a very long time. But they're still fertile and carbon rich to this day. So studies started, particularly a large number of greenhouse studies looking at animal crops. And in tropical areas, they found really positive benefits with biochar. It's less conclusive in temperate soils. Most of the studies have, as I say, looked at crops, but there was a very interesting pot trial that looked at trees. And Swart and Kim inoculated some ornamental trees with Phytophthora pathogens. Now, Phytophthora causes, I don't know if any of you grow tomatoes, and if they're out in the rain, they get that brown tomato blight. That is a Phytophthora pathogen. Phytophthora also caused the Irish potato famine, so it's quite a destructive organism. And the fungus on the Ashdown disease, it might be that biochar added into the soil could help counter that, because Swart and Kim found that biochar, when added at 5% into the soil in pots, reduced disease severity significantly. So it's possible that biochar might have a positive effect on Ashdown, but studies of European trees, so apples in orchards looking at replant disease, have been a lot less conclusive. Now, the other material I was going to test was willow wood chip. So when you do an experiment, you usually have a control with no treatment. In biochar trials, it's often recommended that as well as looking at the pyrolyzed material, that you look at the underlying feedstock to see whether any effects observed are due to the pyrolysis process. In this case, the biochar I was using was made from willow wood chip, so I also wanted to trial willow just as a soil amendment. But willow in its own right is quite an interesting material. So it contains acylic acid, which is the stuff you get in aspen. But it's an important plant hormone, and it's associated with plant defence responses. So the systemic response, whole plant immunity. So willow wood chip in its own right might have a positive effect on the Ashdown pathogen. So to test these hypotheses, I set up two trials, one here at Sunny Ryton and another down in Horham in Sussex. So I studied mature trees at Ryton and in Sussex to have a look at what was going on in the soil and whether that was affecting Ashdown. And I also set up pot trials at the two sites and a field trial. So I think most of you probably saw all the hundreds of little pots out of the back of the building here. That was my Ashdown pot trial, where I tested 13 treatments and a control. There were 10 replicates of each. And I tested the biochar at different volumes, compost, the peat-free compost on its own and mixed with biochar, and the willow wood chip at 10%, both incorporated into the soil and as a mulch on the surface. Then we also tested biochar primed with concrete tea as a nutrient solution, because some studies had found that by priming the biochar with nitrogen, phosphates and potassium, that had a better effect in reducing disease damage. And the saplings were very kindly grown by Francis, who had 2 ash trees in his garden and collected seeds from them and planted them 2 years earlier. And very fortuitously, they sprouted just as I needed. It's almost like it had been planned. So we had the seeds from 2 different mother trees. So I repeated the experiment with the 2 different sets of seeds to see whether there were any differences. So there were 280 pots in total. I also had a site down in Sussex, as I mentioned, and I did set up a pot trial at this site using the clay loam soil there, so to distinguish it between the sandy soil here at Brighton. But unfortunately, even though it was inside the electric net fencing, rabbits got in and scoffed the whole lot, and it's too late to re-establish it. So this is a field trial, hopefully protected by the electric net fencing. It didn't always work, but we also had little tree guards around everything. So the field was laid out in 4 blocks down the gradient of the field, so I could create what's called a randomised complete block design. And that aims to take into account the variation in conditions down what is actually quite a large field. And then within each of those blocks, I had 4 treatments and a control that were placed completely randomly. So again, the biochar at different volumes and willow wood chip mixed in. I couldn't trial all of the amendments because the field simply wasn't big enough. And there were 16 trees planted in each of the plots, and they were repeated across the field. So that gave a total of 320 trees that I would assess. Then around each of these plots, we planted another 20 ash saplings, and that was to try and stop the soil amendments leaching between the different plots. It did have 2 metres between each one, but it's recommended that you do that. So a total of 720 trees were planted, and these were gathered in, you can just about see the woodland in the background, and they came from 3 different locations within the woods. So they had a different provenance, so they're likely to have been living for the first few years of their life with different conditions. So that was the field trial. And then in addition to that, you can see here the trees after 3 years, so this is in 2023, but the trees had done pretty well. They were very slow to get going. I was very concerned I wasn't going to have anything to measure or results to look at, but they did eventually set off and have varying degrees of dieback. The site is surrounded by ash with dieback. So I took soil samples in 2021 from the field trial, and I disease scored the trees and measured them each year from 2021, 22 through to 23. So I had quite a bit of data to work with there. So the data gathering involved measuring the saplings, but also scoring them for ash dieback disease. And the system I used was one that had been devised at the John Innes Centre over in Norfolk, where I had some training with them over there. And it goes from 0 to 10. So 0 is no disease symptoms at all, through the trees with slight disease symptoms, and all the way through to trees that were completely dead from ash dieback, so a score of 10. And that's quite a common method in plant pathology. So the other thing I did was to look at mature ash trees. Unfortunately, we have a few on the site here in Brighton, and there are also a large number at the site in Sussex. So I assessed 130 ash trees in total for size and dieback symptoms. I then took soil samples from a subset of 30 of those trees split between the 2 sites. And I also looked at the ground around each of the trees to see what sort of ground cover and what percentage of ground cover those trees had. And as I say, I took soil samples from the wall to have a look at in the lab. So the things I looked for, predominantly soil moisture, because we've seen that that is likely to have a big effect. And that was done in situ with a wet meter to take readings. Then the soil samples came back to the lab here and were processed, and I looked at the extractable macro and micro elements, so the compounds that the plants need to thrive and grow. I also looked at total carbon, nitrogen and their ratios. And then soil organic matter, because again, that had been shown to have an effect on ash dieback, soil pH, and soil respiration. So I looked at respiration that burst after 24 hours, and also the amount of carbon dioxide that accumulated from those microbes breathing over 22 days. And that's a proxy for microbial activity, so the amount of CO2 that those microbes are releasing. And as I said, I looked at the percentage of ground cover, whether there were forbs and grasses, moss, whether there was impermeable ground cover or bare soil. So we now come to what I actually found from the PhD. So for mature ash trees, soil-related factors did significantly, statistically significantly, influence ash dieback severity. However, the results weren't consistent between the two sites, and they weren't consistent across the years. Now, it might be I only took the soil samples the once, so the soil samples taken early on are likely to have been more closely related to what was going on at that time. But overall, I found that in the sandy silt at Brighton, higher total carbon and higher total nitrogen were associated with increased disease scores in the first year I assessed them, 2021, whereas higher extractable manganese and higher soil moisture and dry conditions were associated with higher ash dieback disease scores in 2023. Whereas in contrast, in the clay soil in Sussex, a higher carbon to nitrogen ratio and higher overall nitrates in the soil, there were lower disease scores. So the soil texture seems to have played an important role in what was going on, that interaction with the tree. Now, it might be that the effects were on the fungus, but it might also be that something is going on between these factors and the ash tree and how it's able to respond, how much energy it has to deal with the disease. It's also likely that the differences in soil texture played a very important role. But because I only looked at the two sides, it's not possible to really draw any firm conclusions. I can't make any sort of firm hypotheses on that. So another interesting finding with regard to the ground cover, at the Sussex site, ground cover did not interact significantly with disease scores at all. Whereas here at Brighton, the percentage of moss cover, as moss cover under the trees increased, so did disease scores. So something is going on with moss ground cover and ash dieback. Now, it could be that as the trees get more diseased and their canopies get thinner, more light can get through, and that might be creating conditions that are conducive for moss. It might also be, though, that the conditions that moss like, so perhaps quite damp, humid conditions, are also really good for the fungus. Maybe the fungus is interacting with moss. It's something that I found really interesting. There are two other studies that have found similar results with moss. Now, I have to be really careful because what I don't want is people going out, finding ash trees and scraping all the moss off the ground, thinking that's going to reduce the disease. But I think this is certainly something that is worth looking into. So for the juvenile ash saplings in the pot and field trial, again, there were effects of the soil-related factors and an interaction with the disease scores, but they were very inconsistent. So in the pot trial, there were a number of factors that influenced disease scores, but the only ones that were consistent between the 2 years was the association between soil organic matter and disease scores. So we are seeing a bit of a pattern which confirms what has been found generally in the literature, that disease is higher when soil organic matter is higher. Now, again, it's unclear whether that is an effect on the ash tree or whether that's an effect on the fungus and increasing sporulation. I think for the juvenile trees, it's more likely to be an effect on the tree because the trees are the saplings are relatively close together. So if there's more fungus on the ground putting out spores, it's likely to affect a larger number of trees. The other factors that were important, electrical conductivity, nitrate and mineral nitrogen, as they increased, disease scores were also higher. Now for extractable calcium, that was one of the few where there was a consistent pattern between the pot trial and the field trial. So higher extractable calcium levels were significantly associated with higher disease scores. And similarly with soil moisture during wet conditions. So if you recall with the mature trees, it was wet moisture levels during dry conditions. So in the summer, probably while the soil is moist, it creates better conditions for the fungus to sporulate. Whereas in the juvenile trees, very wet conditions during, say, rainfall or when the soil was already saturated was associated with high disease scores. So I think what was probably happening here is that the saplings in very waterlogged soils were coming under a lot of stress. So they didn't have as much energy to withstand the disease. So extractable magnesium, phosphorus, total nitrogen, pH, as they increase, disease scores were also higher. Interestingly, in the pot trial here in the sandy soil, extractable manganese levels, as they increased, disease scores were lower. So that's something that contrasted again with what was going on in the mature trees. So it looks like the effects on mature trees of soil-related factors vary compared with those of the juvenile saplings. It's likely that the juvenile trees are having to take up a lot more nutrient from the soil. They're much more reliant on what is going on in the soil at that time. Whereas the mature trees have taken up nutrients, and trees cycle nutrients within them. They're very different to annual plants. So they will have a lot of the nutrients that they need within them. So they're probably taking up and are less reliant on the soil. So that's what was going on with the juvenile saplings. Now, coming to the soil amendments, I know you've all been waiting with bated breath to see what happened. Sadly, there were no significant findings. I think that is perhaps sometimes the case with biochar. So there were no significant effects of any of the soil amendments on Ashdown disease scores. The volume of biochar and the type of soil amendment was not important. The soil amendments, however, had no impact on growth, so they didn't harm the trees, so they still grew and they still expanded in diameter. So I think what we're probably seeing is one of two things. Different biochars respond very differently in the soil, and willowwood biochar might not have been the best choice. It doesn't always have a particularly beneficial effect, and there might be other wood types or other materials that might have had a different effect. But because we're taking a sustainable approach here, willow grows prolifically. You know, I think we've got tons of willow on this site, so it's readily available for farmers and landowners to be able to coppice and convert into biochar. So whatever material we're using, it needs to be something that landowners can readily access and is not too expensive. So the other thing might be timing. So the trees, they were tiny when I planted them, and some of them were still quite small even at the end of the trials. And it might have just been too soon to observe any effects. So studies that looked at apple replant disease and biochar, those that went on for longer than 3 years did find some significant results by the 4th year. The growth was starting to improve. So it might be that it's just trees take an awful lot longer to show results than we see in annual crops. So the other thing that became apparent was the position of the trees within the blocks in the field. That did have a significant effect on disease scores, but only in the 3rd year. So saplings that came from the different provenances, the provenance of the tree had a significant effect on Ashdown disease scores in the first 2 years. So we transplanted the trees from the woods into the field, And for the first 2 years, that position those trees came from was important for ash dieback severity. Then by the 3rd year, that ceased to be the case. And there was no significant difference by provenance, but position within the field had become important. So I think what we're seeing is a time lag between the effects of the soil starting to be revealed in the trees. So the block positions in the field did reflect significant variation. So block 1 had higher moisture readings than block 3, and the soil pH, mineral nitrogen, total carbon, soil organic matter went in a gradient from being the highest at the top of the field down to the bottom of the field. Now, the trees that were tallest were in block 1, and they also had the highest disease scores. And across the bottom field trial, trees that were taller had higher disease scores. Now, you might be thinking that's actually not that surprising. If a tree's bigger, it's got more leaf surface area for the fungus to land on, so it's likely to get more disease score. But if there are factors that are increasing the speed and capacity of ash trees to grow, is that in turn mean the trees are more susceptible to dieback? So do we want trees that are actually going to grow more slowly and then take off? It's difficult to tell because as the trees become infected, they have less resource because the dieback is starting to kill them off, so that their growth is likely to slow down at that point. So that was an interesting observation, but I can't link it in any way to the different soil amendments because each block contained each of the soil amendment treatments. So, in conclusion, and in terms of the implications of my PhD, I would not recommend growing ash saplings with willowwood biochar or the other soil amendments. I think the increased soil organic matter from compost is probably detrimental to the ash tree and gives it more dieback. So I wouldn't suggest we use biochar to remediate ash dieback. Potentially other biochars may be useful, but not willowwood chip. However, in terms of future plantings of ash trees, given that they're now trying to breed ash trees that are more resilient, we probably want to be looking to plant those trees out in areas where the soil is not prone to waterlogging and isn't particularly moist. Ash is renowned for being able to grow well in moist conditions, but it does increase the risk of ash dieback. We're probably wanting to look at soils that have slightly lower pH, but within the ash tree's overall pH tolerance. And we probably want to be looking at more mineral soils, so soils that have lower organic matter. Higher total carbon and nitrogen might well support the trees, and somewhat lower calcium levels in the soil than those we've got here at Brighton and the site at Sacred Earthmead, Sussex. And I definitely think that further analysis of manganese and calcium and their role, and perhaps the difference in their effects on mature and juvenile trees, warrants some further research. So it just leaves me to say thank you ever so much to my funders, who are Lobbs Charity and Coventry University, and also to my supervisory team, Francis, who was my Director of Studies, Liz and Katerina. And thank you all very much for coming along and listening today. So I think we probably have some time for questions. Silence.
So probably a silly question.
No,
Did you know definite whether Francis's trees had ash dieback?
I think they probably did, but they weren't that badly off, because I think they were felled for other reasons than the fact that they.
You mean the parent?
Yes.
Yeah.
Yes.
Yes, you knew, or yes, you didn't know.
Yes.
Yes, they
Yes,
were
they
felled.
have.
They did have.